Hello children, I hope you're having a great day. I hope you're staying safe at home. So here I am again to continue with the photosynthesis chapter. So coming to the last part of the photosynthesis chapter, today we are going to discuss the various experiments that are based on photosynthesis. Okay, so you know for photosynthesis, chlorophyll is required, carbon dioxide is required, light is required, and oxygen is given out. So by various experiments, we will prove that these four factors are required for photosynthesis. So whenever we perform any experiments on photosynthesis, the first thing we need to do is discharge the plant. Now, what do you mean by discharging a plant? It means to remove the starch or food that is present in the leaves. So how do we discharge a plant? We simply keep the plant in a dark room for 24 to 48 hours. So within that time, what happens? Photosynthesis does not take place, and whatever food was present in the leaf will be carried by the phloem to all different parts of the plant. So that means the leaves will be starch free. Now remember, we are performing experiments on photosynthesis, so we need to make the leaf starch free. Why? Because during photosynthesis food will be produced, and that is what we are testing the leaves for. Okay, now the next thing that we do is the iodine test. Okay, iodine test to the leaves. So when we do iodine test to the leaves, what are we doing? We are testing a leaf for starch, whether the leaf has starch or not. So for doing that, what do? We take a leaf either from outside in the sunlight or from the dark room, and we put it in boiling water. Okay, we place it in boiling water. We are placing it in boiling water. Why? To kill the cells. Now remove that leaf from the boiling water and take it straight over to a water bath and put it inside a test tube which contains methylated spirit. So there we put the leaves inside the methylated spirit. Okay, now this test tube with methylated spirit is placed in a water bath. So when we are talking about a water bath, we are talking about a beaker which contains boiling water, okay, which is placed over a tripod stand, and there is a Bunsen burner to boil this water. So why are we doing this? We are placing the leaves inside methylated spirit to remove the chlorophyll. Chlorophyll is removed. Okay. Now, after that, what do? We remove it from the methylated spirit, this leaf, and then when we look at the leaf, it becomes hard and brittle. Now that means it becomes very dry, like dry leaves. Okay. If you place dry leaves in your hands and crush it, you can see that it will easily break into small pieces. That means it is brittle. Okay, so you take that leaf and place it in hot water. Why are we placing it in hot water? To make it soft. Okay, now you take that leaf, you place it over in a container, and then what do? You pour iodine solution over it. Iodine solution over it. That is the iodine test. Okay, now iodine solution is made up of 0.3 grams iodine, 1.5 grams potassium iodide, which is mixed in 100 milliliter of water. Now, after pouring iodine, okay, in the leaves, there will be two results. One result is for a leaf which you have taken from outside, that is, which already contains food. So you will see when you pour iodine solution over it, that leaf will turn blue black. This is not exact color of blue black. Okay, it is something a dark blue, something like the color of your gym. Okay, blue black in color. That means this is the color that you'll get of a leaf which contains starch after pouring iodine solution on it. What does it turn? It turns blue black in color. Okay, why is it turning blue-black in color? Because it has starch in it. Okay, now supposing you had taken that leaf from the dark room and immediately performed iodine test, you know that leaf does not have any starch in it, so you will see that that leaf will turn brown in color. Does not turn blue-black, but what does it turn? It turns brown in color. So if it is turning brown in color, that means it does not have any starch in it. So the two main steps that you have to follow on any experiments on photosynthesis is, first is discharging and then is the iodine test. Okay, so the first experiment is to show that chlorophyll is necessary for photosynthesis. Okay, now for this you have to take a plant which has variegated leaves. Variegated leaves means there are chlorophyll only in some parts and some parts do not contain chlorophyll. Okay, examples of variegated plants are coleus, croton, geranium. Okay, so here we have taken a plant which has chlorophyll only in this region at the center. So what do? The first thing, you place the plant in the dark for 24 to 48 hours. Leaves become starch free. Then you perform the iodine test. That is, you take the leaf, put it in boiling water, kills the cells, then put it in methylated spirit over a water bath to remove the chlorophyll. It becomes hard and brittle. Then you put it in warm water to make it soft, and then you pour iodine solution over that leaf. So what will be the result? You will see that the portion inside which contained chlorophyll, this portion contained chlorophyll, it will turn blue-black in color. Okay, and the portion which did not have chlorophyll did not perform photosynthesis, because chlorophyll is necessary for photosynthesis. So this portion does not have starch. Okay, it does not have any starch. So the portion which had starch turned blue-black in color. Next, we come to the second experiment to show that sunlight is necessary for photosynthesis. So first thing what you do is take a plant, place it in the dark for 24 to 48 hours. Now, before you take this plant out in the sunlight, what do is you take a black strip of paper. Okay, you take a black strip of paper and then you cut some design. Here you can see a star design is cut. You can cut any design. So this is totally black. Why are we covering this portion of the leaf with black paper? To cover out the sunlight. Because what are we proving? We are proving sunlight is necessary for photosynthesis. Totally. So a star shape is there. Okay, you can see. So this middle portion is exposed and these two outer portions are also exposed here. So this part is exposed to the sunlight. This part is exposed to the sunlight. This part is exposed to the sunlight, and the other portion which is covered by the black paper will not receive the sunlight. Okay, so now after doing that, you take the plant and place it in the sunlight for a few hours. After that, you pluck this leaf and perform the iodine test. Put it in boiling water, then put it in methylated spirit, and then put it in warm water, and then pour iodine solution over it. And what result will you see? You will see that the portion which was exposed will turn blue-black in color. Okay, because it had starch. Okay, it will turn blue-black, showing the presence of starch. That means the portion which was exposed to the sunlight performed photosynthesis, and the portion which was covered by the black paper did not allow sunlight to fall on that portion. It will turn brown. Why? Because it could not perform photosynthesis due to the absence of sunlight. So the next experiment is to show carbon dioxide is required for photosynthesis. The very first step, what do we do? We take the plant, we place it in the dark for 24 to 48 hours to remove the starch. Then in the dark room only, you set up the experimental setup in this manner. So you can see that a conical flask is taken or a bottle is taken, inside which we have placed potassium hydroxide. This is potassium hydroxide. What does potassium hydroxide do? It absorbs all the carbon dioxide that is present inside the bottle or the flask. So there is no carbon dioxide inside the bottle or inside the flask. So we take a cork and put it on inside the mouth of the conical flask, and through a small cut we insert a leaf halfway, okay, inside the conical flask or inside the bottle. So this portion of the leaf which is inside the conical flask or inside the bottle will not receive carbon dioxide. So now what do? We take this experimental setup and put it in the sunlight for a few hours. So after a few hours, you perform the iodine test on the leaf that is inserted inside the bottle or the flask. Okay, so after performing the iodine test, you know how to perform the iodine test. Put it in boiling water to kill the cells, then put it in methylated spirit to remove the chlorophyll. It becomes hard and brittle. Then you place it in warm water to make it soft, and then you pour iodine solution over it. So when you pour iodine solution over it, you will see the portion of the leaf which was inside, okay, which was inside will turn brown in color. Why is it turning brown in color? Because it did not receive any carbon dioxide, and the portion which was outside will turn blue-black in color. Why is it turning blue-black in color? Because it received carbon dioxide and it performed photosynthesis. Clear? Now we come to the last experiment, that is to show that oxygen is produced during photosynthesis. Okay, so for that, what you have to do is you have to take a beaker with water and then place some hydrilla plant in it. Now hydrilla plant is a water plant. Okay, that means it lives in water. And you take a glass funnel and invert it over the hydrilla plant. You can see how it is done here. Okay, and then you take a test tube and invert it over the glass funnel and fill the entire setup with water. Okay, now take this setup and place it in the sunlight. After a few minutes, you will start seeing bubbles arising through here. So what are these bubbles? These are oxygen gas that is being produced due to photosynthesis. Now as the gas bubbles rises, there is no place for it to go. So what happens? Vacuum will be created here. That means empty space will start forming at the top of the test tube. What is this empty space? It is nothing but oxygen being formed. Okay, so it will deposit here the oxygen, it will get filled up in this space. So what will happen? The water level will start dropping down. So let this take place for some time, and when the test tube becomes empty, that means when all the water has been pushed down, you test for oxygen. You know how to test for oxygen. You take a glowing splinter near the mouth of the test tube, and the splinter will burst into flames, hence showing that the gas that is inside the test tube is nothing but oxygen. So this proves that oxygen is produced during photosynthesis. I hope these experiments are clear to you.